Okay, so we're starting up now with our next, actually two chapters in this one lecture on cell division. The way I do this is that I combine chapters 10 and 11 from the Raven textbook, the one in the bookstore, just because they are very much related. I didn't really make two separate PowerPoints, I kind of lumped them together into one, and you'll see as we go along, there's a good reason for that, that there is quite a lot of similarity between these two processes. So, kind of the first thing to look at here is why cells need to divide in the first place. And so there's really only about three reasons why a cell would need to divide. The first thing would be growth, right? So all of us started off as a single-celled zygote, and after many millions of cell divisions later, we're now made up of a much larger organism than we were when we were zygotes. And the way that we did that was by through that cell division process. Second function of cell division is to allow for repair. So any sorts of wear and tear, even if it's daily basis kind of stuff, when we lose skin cells, we lose epithelial cells that line our mouths and our stomachs and our intestines, those cells always have to be replaced. Obviously, much more severe kinds of things like cuts or scrapes or burns, those sorts of things. Cell division will also help us to repair the tissues that have been lost that way as well. And then finally, reproduction. And that really, for our purposes, is going to get us more into the realm of sexual reproduction and the animal body. But that's also kind of our third function here of cell division. So, before we get too much into the actual division process, we are going to talk about the life of the cell. And so, cells have what's called a cell cycle, which essentially includes everything that goes on during the life of the cell from beginning to end. So, from the time that a cell comes into existence, all the activities that it does during its lifetime, and then eventually when it either dies or divides, all of those things, all those activities are part of that cell cycle. Right? So there's a lot of things going on here. There's growth, obviously. There's going to be some DNA replication, like we saw back in our previous chapter, chapter 14, when we covered our DNA replication. There's also going to be all the day-to-day functioning, so making proteins and making lipids and detoxifying stuff and building all sorts of things, dealing with the membrane, interactions with the environment. All of those sorts of things take place during the cell cycle. So one of the things that I really like to emphasize in this chapter is that, yes, cell division is an important part of the cell cycle. It's a necessary thing for our bodies to function and continue to function, but it's not the be-all and end-all of a cell's life. Cells may spend, only 5 to 10% of their lives in cell division. The other 90 to 95% of their lives are spent doing other kinds of things, normal day-to-day activities that help to support our tissues and organ systems and really just keep us alive. With asexual reproduction, this is where reproduction occurs from a single parent. And so there is, a degree of genetic similarity that occurs here with asexual reproduction. There's lots of benefits of asexual. Number one, you don't have to find a partner. So for species that live in places where they don't really come across other members of their species that often, asexual is a pretty good way to go. If you're a tapeworm living inside the intestine of a cow or something, or a human, you may not come across a partner that often, and cloning yourself asexually might be a pretty good way to go. You also don't have to worry about mixing your genes together with that of a mate. From a strictly natural selection standpoint, passing on 100% of your genes by cloning yourself is a fitter strategy than passing on only 50% of your genes when you combine them with that of a partner. Now, sexual reproduction does have benefits, right? There's a lot of diversity included with sexual reproduction. We'll talk about later on in this lecture. But with asexual, which is kind of our focus for right now because we're going to start off dealing with binary fission in prokaryotes and then mitosis in eukaryotes, there are several different kinds of asexual reproduction, right? The pictures up here at the top of this slide, these are called hydras. They are animals most closely related to things like jellyfish and corals and anemones and those sorts of things. This picture here is showing hydras that are budding. And what that means is that this adult hydra has all of a sudden a juvenile hydra kind of sprouting out of its side. And this juvenile hydra will eventually break off, and it will go on its way to become a full-fledged adult hydra. So that form of asexual reproduction is called budding. The trees in this picture are going through also asexual reproduction, where we've got this group of trees up here on this hillside. This is actually one genetic organism. If you look at the patch of yellow trees, that's also one organism, and the patch of green trees is again a third complete organism. Which seems kind of bizarre, but if you were to go in and take a DNA sample from any one of these individual trees, from this entire yellow group, or the entire group that's lost its leaf, or the entire green group, Any of these individual trees here would have the exact same DNA as every other tree in this grove of aspen trees. And that's a form of asexual reproduction as well, where they're sort of spreading really underground and then spinning up new stems or new shoots above the ground from there to colonize the land. There are different kinds of life cycles in the living world, right? So if we look at things like protists, algae, and fungi, they tend to focus more on a haploid life cycle. So in these next few chapters, we're going to spend quite a bit of time talking about the terms haploid and diploid. We've already introduced those terms back a couple of chapters ago, but just to kind of review, a haploid cell is one that has a single set of chromosomes. So for example, in humans, if we're talking about a haploid cell, that cell with a single set of chromosomes would have a grand total of 23, because in our species there are 23 chromosomes per set. If we're dealing with a diploid cell in humans, that diploid cell would have two sets of 23 chromosomes for a grand total of 46, right? So haploid and diploid are very important terms to know. So for protists, fungi, and some of the algae, they actually emphasize more of a haploid life cycle where they spend the vast majority of their lives in a haploid state. They become diploid for only a very short period of time before going back to being haploid again. Versus animals, our life cycle over here, we spend the vast majority of our lives in a diploid state, and our haploid stage is only our gametes, our sex cells, or sperm cells and egg cells. Those are all haploid cells, but they're all single-celled, and they're relatively short-lived compared to the rest of the organism. Right? So most of the organism is diploid, and the only haploid cells in an animal life cycle are the gametes. If we look at plants, plants illustrate a phenomenon called alteration of generations, where they actually go back and forth more or less 50-50 between a haploid stage, which is multicellular, and a diploid stage, which is also multicellular. So plants actually have the most complex life cycle, and if you take Principles 2, we talk about that life cycle quite a bit as far as how they go back and forth between being multicellular haploid and multicellular diploid. All right, so just a few slides here illustrating in this case, a protist life cycle where we have a primarily haploid stage with lots of essentially asexual reproduction, and then eventually go back up here to a diploid stage where we have something which is an N plus N sort of stage, and then we go back to being haploid relatively quickly after that. Versus plants that are sort of 50-50. In the plant life cycle, the spores all the way up through a structure called the gametophyte are haploid, gametes are haploid as well. And then we get to a diploid structure, which is, again, multicellular. In the case of a fern, we have a zygote forming on the underside of the structure over here, and eventually growing as a fern would normally appear out of the soil. It eventually looks like this. So in this life cycle, all of this is diploid, and then eventually we get back to being haploid. In animals, we have a haploid zygote, fertilized egg, which goes through many rounds of cell division to create a diploid embryo, which is multicellular. And then more cell divisions, haploid infant or baby, which is multicellular, diploid, I said haploid, I'm sorry, this is diploid up here as well. And then diploid adults through more mitosis, and then eventually meiosis produces haploid gametes, which are going to restart the next generation and form the next diploid zygote. All right, so our next step is to go ahead and look at prokaryotic cell division and take a look at something called binary fission. All right, so right off the bat, binary fission, you can really get a good sense for what it means just by breaking down the term. Binary means two, and fission means splitting. So binary fission literally means these cells split in two when they reproduce. So for prokaryotes, bacteria and so forth, archaea as well, they do not ever have sexual reproduction, because to have sexual reproduction, you have to have gametes. You have to have sperm cells or egg cells or something like that, and because bacteria are single-celled, obviously they're not going to contain sperm cells or egg cells because they are only one cell. They can only reproduce asexually. The other thing about prokaryotes that we need to kind of think back and remember is the fact that prokaryotes always have that single circular chromosome. They never have 2 or 4 or 10 or 20 or 46 chromosomes. They have a single circular chromosome that they have to replicate prior to their cell division. So before they go to actually initiate binary fission, the first thing they have to do, just like we have to do before we go through mitosis, is that they have to copy their DNA. So they're going to copy that single circular chromosome, starting at what's called the origin of replication. And essentially, if you think about a circle, and you imagine starting at a point on the circle, maybe at the top, if you were to copy the circle in both directions, both counterclockwise and clockwise, eventually those 2 points of replication would meet together maybe at the bottom of the circle. And that's really what bacterial DNA replication looks like, is they start at one point, the origin of replication, and replication occurs in both directions until eventually the replication sites meet up at the other side of the chromosome, at which point you now have two separate chromosomes. We then have the moving of the two chromosomes apart from each other to opposite ends of the cell, and then eventually we see a septum forming between the two chromosomes, which will then split that cell in half. So a septum is a really good term to know biologically. A septum is always a divider of some sort, and so in this case, the septum is a cell wall divider, which shows up between what will eventually become two separate cells and split this one large cell with two chromosomes into two smaller cells, each having a single chromosome. So this is what it looks like, right? We've got our bacteria up here, it's got a cell wall, it's got some cytoplasm, it's got some ribosomes, it's got some DNA. And so we're going to start replicating this DNA at the origin of replication, and we're going to create two replication forks, right? One over here, one over here. And then those replication forks are going to move away from each other until eventually they meet at the termination site. Once that occurs, the two chromosomes separate. They are going to move to opposite ends of the cell. And then before too long, what's going to happen is this new cell wall, septum, is going to form between the two chromosomes until eventually we now have two completely separate cells. And at that point, two identical daughter cells have been produced from a single individual cell. And again, because this is asexual reproduction, there is absolutely no diversity here. Aside from any mutations that might occur, there is no genetic diversity. These two daughter cells are exactly the same as each other, and they're exactly the same as the parent cell that started off this whole process. So no genetic diversity happening here. So from here on out, at least for the chapter 10 part of things, we are going to be looking at mitosis. All right, so mitosis is defined as being nuclear division. All right, so before we get into the slide, let's talk about a couple of terms here. Mitosis is nuclear division. So if you could have a cell that just went through mitosis but didn't do something else called cytokinesis, you would literally have a single cell with two nuclei, because it went through mitosis, which is also nuclear division. The nucleus divides to form two nuclei during mitosis. Cytokinesis then takes that one cell with two nuclei and splits it into two daughter cells that each have its own nucleus. All right. So getting back into some of the stuff that we talked about in the last chapter. Remember in the last chapter we started off with chapter 14, and then we kind of jumped into chapter 10 for a little bit, and then we came back to chapter 14. This is kind of going back and reviewing a little bit of what we saw before. Prior to DNA replication, each chromosome in a eukaryotic cell consists of a single structure. After DNA replication, we now have those two sister chromatids. So again, let's relate this back to humans. Humans have two sets of 23 chromosomes, one set from mom, one set from dad. Grand total chromosomes then is 46, so two sets of 23 is the same as saying 46 chromosomes. After DNA replication, all 46 of those chromosomes are going to exist as identical copies. So you actually would have two of your dad's chromosome number 1, two of your mom's chromosome number 1, two of your dad's chromosome number 2, two of your mom's chromosome number 2, and so on. So even though you'd have 46 chromosomes, you actually would have a grand total of 92 chromatids. And those pairs of sister chromatids are always held together in the middle at the centromere region by those cohesin proteins. And we talked about those a bit before as well. Cohesin proteins are what actually join the sister chromatids together. So again, putting this back into the big picture sort of scale of things, why do we want to have two copies of every chromosome? Well, eventually we're going to try to produce two daughter cells that are completely the same. And unless we have two copies of every chromosome, there is absolutely no way to make two daughter cells that are completely the same. So prior to cell division, we have to copy the DNA first, and then, and only then, can we divide that DNA evenly into two daughter cells. Okay? So we've taken our DNA, our 46 chromosomes, we've copied all 46 of those chromosomes. Now we have two copies of each of the 46 chromosomes. All right, we also talked about this in the last chapter as well. DNA packaging, right? DNA, we said, in the double helix form is way too long and way too uncompact, too stretched out to be able to fit inside of a cell. So we take our DNA and we wrap that up around what are called histone proteins to create what are called nucleosomes. So nucleosomes represent, again, DNA wrapped up around histone proteins. We then take those nucleosomes and wrap those up into a coil called a solenoid. And solenoids, again, we talked about this before, this is the form that we find our DNA in most of the time in our cells. We never find DNA in a human cell that just looks like a double helix because there's no way it would fit. It's way too long to fit inside of a cell. And similarly, we also don't find our DNA in a nucleosome form in our cells either because, again, it's way too uncompact to be able to fit inside of a cell. So once we get to the solenoid form, then we have DNA which is actually able to fit inside of a nucleus and still be read. It's still expressible DNA, even though it's been packaged up into a slightly more condensed form, or a very much more condensed form, actually. Eventually, we take those solenoids and we wrap those up around scaffold proteins, and that creates this kind of a structure, which is normally, when we think about what chromosomes look like, it looks something like this. In reality, what nobody ever tells you is that the only time our DNA actually looks like this bottom structure is right before and during cell division. Right? The rest of the time, it's going to be more like this up here. But this is the form that it looks like when our cells go to divide. Again, just getting a quick review here of the terms homologous chromosomes and sister chromatids. Homologous chromosomes are chromosomes that contain the same genes, although the versions of those genes can and very often are different from each other. So if we say this is chromosome number 9 from dad and chromosome number 9 from mom, those two chromosomes are homologous. Both of them contain, for example, the gene for blood type, AB, or O blood type. AB also is an option there. So because they both contain the blood type gene, we know these two chromosomes are homologous. Now, do the two alleles for that gene have to be the same? Absolutely not, right? You could have dad's chromosome number 9 saying, okay, well, we're going to code for the A version of blood type, and mom's is going to code for the B version of blood type. So even though they contain the same genes, even though they both contain the blood type gene, the specific form of that gene, or the specific allele, or the actual version of the gene doesn't necessarily have to be the same between homologous chromosomes. Now, after we go through DNA replication, now we have two identical copies of dad's number 9 and two identical copies of mom's number 9. So these two sister chromatids, not only do they have to have the same genes, but because they're the product of DNA replication, they also have to be completely identical. So if the blood type gene is up here, and this is an A, then automatically this also has to be an A. And if this is a B, that has to be a B, because these two have to be identical to each other. Right? Sister chromatids are always identical to each other. Homologous chromosomes are similar, but not identical to each other. Okay? So, moving on then, we are going to take a quick look here at some pictures of chromosomes. And again, this is in this particular state here, where we have two of each of these structures, right? That's after DNA replication. Normally, we don't find two complete connected chromosomes for all these chromosomes. This is happening only after DNA replication has taken place. So the vast majority of the time, the DNA in our cells is not going to look like that picture. Okay, so as far as the parts of the cell cycle, the eukaryotic cell cycle has several parts that make up the entire process, right? So the first part is called G1, or gap phase 1, or growth phase 1. I've heard all of them used. They're all perfectly fine to use those terms. Growth phase 1 or gap phase 1. This is where a cell spends the majority of its life. Maybe 80 to 90% of its life is spent in gap phase 1. So this is where a cell is going to grow. This is where a cell is going to do all the kinds of things that that particular kind of cell does, whether it's making proteins or making lipids or detoxifying things or consuming debris inside your body, whatever. Whatever the cell is doing for the vast majority of its life, it is doing it in growth phase 1. From growth phase 1, we then go into DNA replication, which is called S phase. The S is short for synthesis, and specifically, what are we synthesizing? It's DNA. So after growth phase 1, the cell goes ahead and copies its DNA through all the DNA replication process steps we talked about back in our previous lecture. From there, we go to a second growth phase, or second gap phase, called G2. And this is where a cell is really getting itself ready for the actual mitotic and cytokinesis process. It's like the pre-flight checklist for cell division, right? So we're going to copy the organelles, we're going to make more cytoplasm, we're going to get our microtubules ready, which if you remember all the way back in chapter 4, microtubules are part of the cytoskeleton, but specifically the microtubules in this context are used as a way to move chromosomes around inside the cell on what's called a mitotic spindle. So microtubules are going to form this mitotic spindle during G2 as well. After G2, we're then going to go into mitosis, or M phase, and then after mitosis, we're going to go into cytokinesis, where we're actually going to split that one cell with two nuclei into two separate cells. All right, so growth phase 1, S phase, and growth phase 2 collectively are all called interphase. And again, like I said before, interphase might be 95% of a cell's life. The remaining 5% of the time, or even less sometimes, is spent during mitosis and cytokinesis. All right, so the vast majority is spent in growth phase 1, but also the rest of interphase. And then for a tiny, tiny little period of time, the cells would go through mitosis and cytokinesis to copy themselves into identical cells. All right, so that's kind of the basic structure of the cell cycle for a eukaryotic cell. Different cells grow at different rates. Different cells will complete the cell cycle in different amounts of time. Embryonic cells, for example, have very, very short cell cycles because For an embryo, the faster you grow, the bigger you get, the faster you get there, the more likely you are to survive. So for embryos, they're going to divide relatively quickly because getting bigger is a recipe for success and survival for those kinds of cells. Other cells take a lot longer. Liver cells, for example, take about a year to get through the cell cycle and get to the point where they actually become functional. Some cells never divide, right? So skeletal muscle cells and cardiac muscle cells and most of your neurons do not divide ever. So they actually are stuck permanently in G1 and they never go beyond that point during the entire time you're alive. So again, that's kind of the overall picture here of the cell cycle, but just reiterating the different cells have different characteristics when it comes to their cell cycle. How long they spend in G1 or G2 or whatever, how long it takes them to mature differs depending on which kind of cell you're looking at. If you're looking at skin cells or stomach cells or small intestinal cells or cells that lie in your mouth, they have a very short lifespan. Stomach cells have a lifespan of 2 to 3 days before they break off and are digested by the rest of your body. So their cell cycle is really fast, versus other cells take either a really long time or in some cases they never divide. A lot of diversity there on the way these cell cycles actually work. So this is kind of a quick overview of the cell cycle. This is a cycle, so there really is no beginning or end. We can pick any point on here and kind of follow it through. So just arbitrarily, I think today I like green, so we're going to start off with G2. G2 is going to lead us into mitosis, which for our purposes, I typically don't cover prometaphase as a separate phase. I just keep it with prophase, metaphase, anaphase, telophase. And then we go into cytokinesis, where the cell splits. And then now we have two cells that each go into their own separate, respective G1 parts of the cell cycle. Eventually, both of those cells get to the point where they reach their S phase, where they copy their DNA. And then both daughter cells go through G2. And then both daughter cells go into mitosis. So the whole cycle repeats itself at that point. Okay, so this is the correct sequence of the parts of the cell cycle. And again, this slide is kind of reiterating what we talked about a couple slides ago. Interphase consists of G1, S, and G2. G1 being the point where the cells actually grow the most and function, S being where they copy their DNA, G2 being where they get ready for mitosis by getting their chromosomes more condensed, copying centrioles, which we'll talk more about a bit later on, getting the microtubules ready, and just generally making sure the cell is healthy enough to attempt to go through mitosis. Centromeres, remember, are the points of constriction on a chromosome. Once we get them into that fully compact form, they have that part in the middle where they kind of squeeze together called the centromere. The centromere is also where we find a structure called the kinetochore. And the kinetochore is a structure where the microtubules will eventually connect in order to be able to move the chromosomes around inside the cell. So kinetochores are a place for microtubules to attach. And then finally, cohesin protein, kind of another term here, is also found in the centromere region, but only when you have sister chromatids attached to each other. Once the sister chromatids separate and move apart during mitosis, the cohesin protein is broken down and goes away at that point. So it's a very temporary kind of a structure in these chromosomes. All right, so we're gonna kind of end things here. We've got a cell. It's an interphase. This is actually an animal cell. It's a cell of a whitefish blastula, which is an embryonic stage in animal development. But for our purposes, what we can see in this cell is that it has a plasma membrane. It has a nucleus. Inside of here, there'd be chromosomes. You really can't see them too well, but there are chromosomes inside this nucleus. There's cytoplasm out here and a bunch of other organelles that we really can't make out because it's fairly low magnification. But that's kind of the basic structure of a cell in interphase, where you can see the membrane, you can see the cytoplasm, maybe you can see some kind of rough outlines of where a few organelles are, and then you can also see the nucleus. After that point, we're going to move into the beginnings of mitosis. And actually what we're going to do is we're going to cut off this section here, and we're going to start up with mitosis and go through the 4 phases, and then also work through cytokinesis. So we're going to cut this section off here, and I will get it posted and start the next section here shortly.